Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this episode is going out on, dare I say it, Friday the 13th of June. Are you superstitious? I guess I'm not, but it's also something that I definitely don't want to test too much. As I was at an event this week, I got the interviews for today edited early. However, that doesn't mean that the entire process was completed earlier, but it did make it a bit easier in theory. Before we get going, if you do have suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com. And that's also the address you can use if you are interested in advertising on or sponsoring one or more podcasts, as that's something that we offer too. And we do have one or two lined up already. I'm mindful of not having this sound like a commercial, so if you do want to be involved, we'd be glad to have you on board. And we're not expensive. Last week, our guests were Ben Cliff, CEO of CRO Concept Life Sciences, and Ravi Rao, Chief Medical Officer at Citrix, a company taking on, among other things, autoimmune and inflammatory diseases. This week, a little later, we will be chatting with Dr. Peter Tummino, President of R&D at Nimbus Therapeutics, in an in-depth discussion about not only what Nimbus is doing, but also on the role of computational chemistry in drug discovery and development. But first, Syngene International Limited is a global contract research, development and manufacturing organisation, or much easier to say, a CRDMO. And it's acquired its first biologics site in the US, fitted with multiple monoclonal antibody manufacturing lines. To tell us about it, what it means for Syngene and its customers, is Alex Del Priore, Senior Vice President of Manufacturing. First, I wonder if you could give me a little bit of background about Syngene.
Thanks, Jim. Over 30 years ago, Syngene started with its journey from humble beginnings as a contract research organization, and we've grown during that period to naturally move to what is today an integrated both contract research but also a CDMO provider. Over 8,000 employees strong, working across a number of modalities in both small molecule and large molecule, spanning discovery, development, clinical supply of GMP material, all the way through commercial manufacturing.
And what are you currently able to provide from your existing sites, and where are those existing sites?
So I'd like to focus today a little bit on our biologics portfolio. So in biologics, we have our discovery business, which is located in India, a number of locations in both Hyderabad and Bangalore. Obviously, we've just announced an acquisition, our third acquisition in the last 18 months in our biologic space that expands our development and manufacturing from beyond the borders of India into the U.S. It's our first asset located outside of India, and obviously an exciting time period for us. A lot of challenges that we are certainly ready to address and take advantage of for our clients and our patients.
And what was the rationale behind moving into the US market?
Jim, when you look at the U.S. market, obviously the largest in the world, growing in biologics terms 10 to 15% per annum, there's a large portion of that market that prefers to stay on, within the country. So we suppliers, CDMO providers, typically deal with those that are wanting to export some of their services to places like India and other low-cost regions. But by and large, the bulk of clients prefer to be in the U.S. And so when we look at our overall strategy, how we're going to grow and serve our markets, it's, I think, a natural fit that we move our assets to where we sell.
Things have changed drastically in the US in the last several months. Has that been one of the reasons, or was it always going to happen anyway?
I think if you refer to some of the tariffs that have been discussed and some of the Biosecure Act that was, maybe over a year ago, you had COVID prior to that. I think what it really revealed was the importance of having a diverse supply chain, one that can function given the uncertainties, which I'm sure we'll continue to have in the future. When you look at whether you're developing a drug or a process, whether you're trying to optimize, whether you're trying to provide those first important GMP batches to support the clinical trials with patients, or whether you're making commercial, you've got challenges in each one of these steps. And we have a philosophy that we've been working towards, which is you want to make where you sell and you want to source where you make. And so if we're going to be a global player, then you need to have a global set of assets. And that's really the underpinning of why we chose to expand at this time into the U.S.
And I guess that makes sense both economically and also in terms of sustainability.
If I could maybe just expand a little more, every client is different. Every client has unique needs. So what we offer today in one word is flexibility. So if you're seeking value and you want to look at what you can do in a place like India, then certainly we can offer that. If you're looking for proximity and speed to market in the region where your activity is, we can now offer that in the US. What I suspect is probably going to happen, though, like always, in any risk mitigation strategy, you're going to select a hybrid. You're going to do some things in India, in a low-cost region. You're going to do some things in the region in which your clinical trials or your commercial product is being sold. So it just makes sense that we provide that flexibility to our client base so that we can serve the customers, which is what we're ultimately about, which is getting affordable medicines to patients regardless of where they're located in the world.
For sure. Yeah. Could you tell me a little bit more about the acquisition itself?
So, Jim, when we looked at this, it was quite a long negotiation, if I must say, but it's something that we've been evaluating for probably the last 18 months. And with this facility, with Emergent, we found the right partner, and we were able to do this deal. It's located in Baltimore, Maryland, at what's called the Bayview campus. It's a great ecosystem that is centered around really three locations in this Bayview campus. One, you have the Johns Hopkins Medical Center, which is one of the leading hospitals for research in the U.S. You also have the NIH office, which supports much of the funding that many biotechs seek as they look for innovation and new drug discovery. And then, of course, we have our facility. So located right in a great location. And then if you go a little further out, it's one of the pharmaceutical biohubs. So the access for talent is always one of the conditions that you look at when you're considering building, partnering, or acquiring assets. And I think we found that in the Baltimore area.
And it's a massive area in terms of population as well. When you consider, Washington, Philadelphia, it's kind of right in the middle of everywhere.
It's in the pharmaceutical corridor, as we call it. Up Highway 95 from Baltimore all the way up through up to Boston. So yeah, certainly a lot of talent there. And I think the universities that support the research, the University of Maryland located in that region has some of the leading researchers, particularly in novel space, whether it be cell therapy, gene therapy, which are all things that we're interested in.
Could you maybe go over one or two of the main advantages that having the facility will mean for the company?
Let me think about that. There's probably two or three, right? So I think the first thing that we've looked at is obviously access to the market in which we sell. Number two is capacity. We've been growing now for the last 4 years, which I've been at Syngene. We've been growing exponentially. We've sold out our first plant. We then had to acquire and add capacity for our plant number 2, 3, 4, 5. This will actually be our suite number 8, 9, and 10 located in the U.S. What it's also added, in addition to capacity, is a different reactor set or reactor options. One of the things when you start looking at different segments in the market, some segments are focused on cost, some segments are focused on time. For those that are cost conscious, the size of the reactor is important. And so typically your single use platforms have traditionally stopped at 2,000 liters. This facility in Baltimore employs the 4,000 liter single-use reactors from ABAC. So that will give us, again, greater flexibility for our clients. Related to that is basically the exploration of the single-use technology. So for those clients that are really looking for speed, this allows you to change over much more rapidly than a stainless steel type reactor. And I think that's, it was novel, quite a few years ago. It's become pretty commonplace. And I think it's proven its worth, demonstrated the ability to bring our clients to that first decision point, which is getting to that first in human trial in phase 1.
What will the benefits be for your clients other than obviously the proximity? And do you think that it will also attract new clients?
We certainly hope so. I think being in the U.S., again, part of what people don't realize when we talk about exporting, some of the services to a place like India or a place like China or some other place in the world, that the lion's share, the majority of the customers, whether it's a biotech or large pharma in the U.S., prefer to be in the U.S. So in the terms of addressable market, roughly 60%, maybe a little higher, 60 to 70% of all services provided in the CDMO reside in the U.S. And a minority share is what's actually exported offshore. So being in the U.S., we have a series of customer days, a series of consultant days that we'll be hosting or have hosted and will host for the rest of this year to invite people to come and see what we have to offer. It's a compelling value proposition.
What kind of advantages will your clients in the US be able to see by having you closer?
If I could, Jim, maybe broaden that out a little more and not just on location. Let me talk about Syngene's capability. Having grown up as a company that invested heavily in science and research, there's a lot of technology and capability that we work on. So many CDMOs may choose to work in a biosimilar market as it relates to biologics or some mAbs. We certainly do that. That's our core business. But we've been successful in leveraging our scientific talent to go into all the novel modalities. So things in the name of, if I throw out examples, ADCs, the full-fledged capability of making a payload, a linker, the mAb, and doing the bioconjugation is something that we provide. If it's in cell therapy, if you're making a CAR T treatment, for example, there's a number of consumables. There's, mAbs that are used. There's the viral vectors, et cetera. We have been working in that space for many, many years. So when you work with Syngene, what you're working with is a scientific company that believes in innovation, that works closely with those very difficult solutions in evolving markets, as well as the traditional monoclonal antibodies. So in doing that, we can provide that service, whether it's in India or whether it's in the US or the hybrid model, providing flexibility for a specific customer who's dreaming about that next innovation or the next cure for the incurable.
Are there any other things that sets Syngene apart from your competitors?
I think the ability to put together a value proposition that meets the need of the specific client. We have a whole platform or a whole culture around customer centricity. And so if you're looking for high volume generic services versus innovation, then you probably might want to select someone else. I hate to admit that, but what you really want to do when you work with Syngene is you want to take advantage of our science, our capabilities, and our ability to innovate in new spaces. We pride ourselves in solving complex chemistry, complex biology. So we're working on bispecifics, trispecifics, a number of different conjugations. So a lot of things. I think we have, over the 30 years, partnered with over 450 clients, ranging from biotechs as well as almost any major biopharma company that you'd be aware of. We've developed hundreds of patents that we provide to our clients as a result of this service. And I think the ease of doing business will become even easier now that we're closer to the client. And you can work in the same time zones as our customers.
What's the reaction been like so far from your US-based clients?
I would say very exciting. It's just an exciting time to be alive anyway. You got cures for things that in our lifetime were not curable. And we're seeing these great innovations happening. Gosh, it just seems like they roll off the printing presses these days with new solutions for patients. As we put together the strategy of making this entrance into the US, we went and talked to some of our key clients. And each of those key clients said, if you do this deal, we want to make sure that we talk to you because we've had our experience, in India with you, but boy, it sure would be nice if we had an asset a little closer to home. And so that's what we do. We have lead anchor clients that we will be bringing in this year to talk a little more about the facility itself. This facility was shut down as part of the emergent strategy change. However, we kept it in warm standby. So we didn't shut down the utilities. We kept the water running. We kept the steam running. We kept the environmental monitoring going because we knew that we wanted to restart this facility quite quickly. And we anticipate that by the end of this year, we'll be starting our first GMP batches. And we'll be doing that with our anchor clients.
Are there any challenges associated with being in the US or newly in the US?
I have to kind of chuckle with that question, right? Aren't there always challenges?
Well, yeah. Specific challenges then, I guess.
Yeah, you could imagine and foresee that a startup of a facility, usually it's about getting the equipment up and operating. Technically, it's about staffing. Technically, for the startup of this, because this facility's already run, it's in really good shape. Emergent did a fantastic job in the shutdown so that it could be restarted quickly. For us, the specific challenge is doing the staffing. So we've already hired, for example, our US HR lead. We have offers out to a site leader at this point, and we will be ramping up the hiring process. The second focus is to take our quality management system and implement it into this new facility. We want to make sure that we are consistent regardless of where we're located in the world. So I would say that it's really those two things. It's about staffing. It's about putting in the quality management system. And if we do that very well, then it's about we're open for business. Please come and see what we have to offer at Syngene.
What are you facing in the next, I don't know, year, 18 months? What's the focus?
What we do, Jim, is if we buy a facility or build a facility, we don't start up the entire site at one time. What we do is we bring it up in phases. And if I talk a little bit about this facility, it has 4 suites. We will bring up the first suite at the end of this year. And then as the demand grows with our client base, we will bring on the second suite, the third suite, and then obviously the fourth suite. So 8, 9, and 10. We call that biologics manufacturing plant 8, 9, and 10. And then we have the development line, which we'll call BMP 11.
Is there anything that you wanted to cover that we haven't talked about?
No, I just think in closing, for those who don't know Syngene, please give us a call. We have quite an offering as an integrated supplier from the very beginning of discovery through animal models, through actually running clinical trials, developing our processes, whether it be in small molecule or biologics, large molecule, going through the development into phase 1, phase 2, phase 3, and commercialization of projects. We do that across all modalities and invite a conversation for those seeking a solution to make their dreams a reality.
That's good. Has AI been any factor in what you do?
When we look at the digital footprint, so let's just talk about AI in the form of a digital footprint. In the service world, one of the things that the clients seek and what they crave is complete transparency and real-time data. And I think if I can put it under AI, it's having that digitized workplace. So for example, all of our batch records are electronic batch records. All of our quality management system is online and in the cloud that any customer can see at any time. When you look at our QC laboratories, which become one of the building blocks of the QMS system, the amount of technology that we have in there using AI that actually modifies and troubleshoots some of the equipment that we use is being employed. We use visual, retina recognition and face recognition so that we have track and trace across our digital platforms. And then as it relates to discovery, we're constantly developing those digital twins and those models that allow us to expand and build on the greatest tool, which is still the tool between your two ears, your brain. Augmentation is a great thing. Technology is a great thing. It enables innovation, and that's what Syngene's all about.
Sounds like a great company making big strides in the US. It's a while since I've been in Baltimore, but it's a nice city. Except when the Orioles are beating my Jays, which is pretty much all the time. But that's another subject. Moving on, but still within biotech, is Nimbus Therapeutics. And we're going to get an in-depth look not only at what the company's doing, but also at what computational chemistry means in drug discovery and development and the biotech industry as a whole. To tell us is Dr. Peter Tummino, President of R&D at Nimbus. First off then, I wonder if you could tell me a little bit about the history of Nimbus Therapeutics.
Yeah, so Nimbus Therapeutics was started almost 16 years ago as a computational drug discovery company by Bruce Booth with Schrodinger. And we started using the physics-based computational tools like FEP with the idea that that could really drive making better or best-in-class small molecule agents. And over time, we have become independent from Schrodinger. Both groups are doing their independent drug discovery, though we do still work with them in terms of the software they offer, which is part of our core. We have kept our scientific core of structure-based drug design. We've added to it with deep expertise in structural biology, molecular sciences, and now are incorporating AI and ML where it makes a difference in terms of developing novel clinical candidates. Nimbus is a private company. We are an LLC model where every discovery development program is a separate company that is part of Nimbus as an LLC, which we could talk about is helpful in terms of when there is interest from a partner, they can acquire a program and Nimbus continues on. They simply are licensing or a different company is acquiring a program. The areas we're in are I&I, oncology, and metabolic disease.
And you were mentioning AI there. You've got a drug discovery engine. I imagine that in 16 years, that's probably evolved quite a bit.
Yeah, it has. It's interesting. When I started 33 years ago, I was in structure-based drug design. And some of the elements are the same, as you know, probably from docking. Docking has moved now to FEP, more physics-based, and it's absolutely more powerful. Some of the elements there are the same. And we think it's probably the most powerful method for making, when you have very high demands on a small molecule, it's probably done better by structure-based drug design. And so that remains our core. In fact, I would say we are much stronger at it than we were 5 and 10 years ago. We've really built that up, much more structural biology as an example of that. But we continue to add. I would say our approach to AI is skepticism, is to start with, where is this going to make a meaningful difference for the Nimbus model today? And it does in certain areas. One of those is ADMET. ADMET predictions, we think AI has made a big difference, and it's completely incorporated into our methods. Generative AI is beginning to have a nice benefit for us. A lot of the other areas we see as still nascent and forming and not necessarily ready to have a major impact on bringing forward development candidates, small molecule development candidates.
Do you think that some companies that see AI kind of jump on a bandwagon that they're not really sure what that bandwagon is?
Yeah, AI is a topic unto itself, or AI in drug discovery. And I've given this some thought, and it's interesting. I think there's examples of platforms and technologies over the last 30 years that make a big difference, but do not, quote, revolutionize the industry. I don't think AI is revolutionizing the industry. People have jumped to simple conclusions that the future of drug discovery is AI. It is not, in my opinion. It is a set of powerful tools that could be useful in the hands of very good drug discovery people. And so I think if nothing else, it's being characterized incorrectly, and that's going to lead to issues and disappointment in results going forward. I think that's happening.
Your own drug discovery engine, how does that work?
Yeah, so there's some essential components, and one I've already talked about. We work on programs where you can take a structure-based drug design approach, right? And what is amenable to that is much larger than it was years ago. And I think the field's gone in that direction. The ability to get high-res structures, either in the lab or computationally, is excellent. And then the ability to, especially if you have a starting ligand, to optimize it is much better. And that's the core of what we do. But what's interesting is what's added value over time is, I would call it proprietary target selection. I see a lot of companies that can drug targets but don't know which ones to pursue all the way into clinical trials. And that's a big focus of us. So the amount of effort Nimbus spends on target selection in our 3 therapeutic areas keeps growing. We think there's really adding value there. There's aspects also of really broad drug development expertise is a key part all the way through phase 2 trials. And then a key element, which you might describe as cultural, is we take pride in killing programs. And so attrition is built into it. Attrition allows you to set high standards, to hopefully do that in an objective manner. The ability to kill programs means you can fully fund the winners. And for a biotech especially, I think that's exactly the right way to do it, is big bets on programs with the most promise. And that's what we do.
Is that part of what's made you so successful?
Yeah, what I just described, I think, is what we would call our secret sauce. Well, one, the ability to look across 3 therapeutic areas. We don't pick that many targets. I think we can pick those. And this is the part I did mention, where what we do, which is our goal is always to make the best in class molecule. We can be pretty picky. We know that's only going to be in a minority of cases. And it's where things set up quite well, where we think there's an opportunity where there's a major attribute that isn't solved regarding the molecules out there, and we can go do it. And it's relevant ultimately to a patient and their treatment. And so that's what we go after. I will say that we've had success. ACC, TYK2, HPK1, WRN, we believe there's a case to be made that we have the best in class molecule for each of those. And we've had some confirmation. That's not to say we haven't had failures also, of course, but we think the model has worked well to focus on making the best in class. And that engine has worked reasonably well for us.
And you've had a couple of fairly big deals. How did those come about?
Yeah, the first one was ACC, acetyl-CoA carboxylase, and this is going back a few years. And we started on that target, thinking of it as a good target for diabetes. As preclinical data began to come out, it became clear that this had much more potential for NASH, and we pivoted in that direction. We made what we think are very good molecules. Interest in NASH went up a lot after Intercept's phase 2b results, and we had entered the clinic. And so this is the first time Nimbus went from pure discovery into the clinic. We pursued that in phase 1 for that execution. So that was a new phase for Nimbus. After that, we had broad interest from large pharma and then ended up partnering that with Gilead for $1.2 billion upfronts and milestones. That currently is in phase 2 trials right now in combination with FXR and GLP-1, and that should be reading out shortly. So that's in a large phase 2b. So that was, I would say, our first big success. The second was on TYK2. TYK2 is a JAK. It's also called JAK4. And people know the history with JAKs, that JAK 1 through 3, JAK 1 and 2, agents that are used today. I think they all have a black box warning as you dose at a high level, some of the AEs you see. The promise of TYK2 is that it would be safe. Therefore, our goal was to have a TYK2 inhibitor that was entirely selective. BMS with deucravacitinib was ahead of us. Theirs is 75-fold selective against the JAKs, and ours is 1.5 million-fold selective. That's from our SBDD approach. We took that into the clinic. We went into phase 2b for psoriasis and for psoriatic arthritis. In psoriasis in particular, we had superior PASI scores or superior responses. To some degree, our agent, the small molecule, looked like a biologic, which was quite exciting. After that, with those results, we received high interest across large pharmas. We received multiple offers. ultimately signed a deal with Takeda for $6 billion in total, and that's progressing through Takeda. It's actually quite exciting, the work that Takeda is doing. They're in phase 3 with the agent and also in multiple phase 2s. So those are two of our big stories, and we push ahead into other programs.
When it comes to working with other companies, do you have a specific strategy? I mean, do you work with them on what they're looking for, or do you work independently and then try and work with the companies afterwards, or does it depend on the company?
We work independently. And so a big part is doing our own target selection, owning the programs, fully funding them, and then pushing them forward at some point. We always ask ourselves as we progress our program, which company is best to shepherd this? And for a good period, that's Nimbus. At some point, it might be better run by a large pharma. And so I would say that's the rule. I can think of two prominent exceptions. And one is Lilly on AMPK, and then previously Celgene on multiple targets. But those are the exceptions. As a rule, we work independently, and then ultimately at some point, we do engage with large pharma.
Speaking of AMPK, I wonder if you could tell me a bit about your work in that area.
Yeah, so we love the story around AMPK activators. These have been pursued for multiple metabolic indications in the past. Our initial interest was in NASH and our goal was to make an isoform selective AMPK activator. And we began this program. What happened as it progressed is there were results that came out in NASH that there was actually a rejection of a trial at phase 3 that really forced us to reflect on our plans and what we needed to do in clinical trials. And so we made one of those hard decisions I mentioned. We stopped the program. As I said, that's common at Nimbus, is to stop so that we can fund the others. And we put it on the shelf. And 6 months later, Lilly approached us. They had heard about our effort and made an offer to do this in partnership. And they had their own plans in terms of how they would take it into the clinic. We've progressed that program. I'm limited in what I can say. I can say that in the Lilly partnership for AMPK activators, we've reached a major milestone. I think that we've had a lot of success on that, and it's actually been a great partnership. So we'll see how that comes out. Hopefully some of that ultimately will come out into the news.
You mentioned a couple of instances where you've been working on one condition or disease and pivoted at some point. Is that common?
It is. It is being responsive to the market and what is there. Multiple examples. I gave you one. We had major efforts in immuno-oncology, and the scientific value for immuno-oncology, the proposal of benefit for patients, I think, is the same, as is the unmet medical need. The investment community is less interested in IO today because of failures. And so this is an area that, for example, we had a CBL-B program that we stopped. There's an example. I mentioned AMPK. There's others where that's exactly the case. They're stopped for scientific reasons. We often put in place, as clear as we can, validation around biology or chemistry, milestones, and we'll stop. But we will respond to market conditions also. And that's actually, I think, been a very appropriate way to do this for a small company that has about 6 targets at a time, 6 programs.
Are there companies, obviously not yours, but are there companies that find the timing of that difficult? I mean, do some companies keep going when they shouldn't?
Probably. I don't have a comment except to say we think a lot about discipline or skepticism side by side with the conviction or the reason to believe in to do it on a target, and lean heavily on skepticism, lean heavily on why this might fail and what might be the difficulties of clinical tractability, of the value of this program. And getting that balance right, I think, is a key to success.
No, it just seems like it's a very good trait to be able to have that capacity to know when to stop or when to pivot to a different disease. It seems to be that that's something that would really benefit a company to have that insight and that ability.
Yeah, you said it better than I did. I agree. I think small biotechs are known for their passionate pursuit of the target. And there's an advantage there of knowing when to have the commitment to stick with it. In discovery, we worked on TYK2 for, I think, 6 years. And so there can be a benefit. You might even say it's the discernment of when to know, when to be skeptical and stop. But there's other times you really do need to stick with it for 6+ years to get there. And you need both elements. And you need them in the company, and you need the disagreements, I think, to get to the right decision.
What other difficult targets are you currently looking into?
Yeah, so I'll mention two that are more advanced. One is Werner's helicase, or WRN. This was identified in 2019 by multiple groups as a highly valued synthetic lethal target for MSI-high solid tumors, colorectal, endometrial, and others. And I know that many companies jumped on it to work on it. There were no ligands at the time. Difficult program to drug. 5 years later, there's now, at last count, as of this month, 4 groups in the clinic with a molecule. And we are one of the 4. We have preclinical data to show that we believe we have a best-in-class molecule, a greater on-target potency that we expect is translating into lower efficacious doses, allowing us to, you might say, hit all tumors most and less sensitive that are MSI high. And so we're in a strong position there. Our first patient dosed was in March, and we're excited about that program. That's a non-covalent inhibitor of WRN. Another program we have whose next target is development candidate is salt-inducible kinase type 2, or SYK2. This target has been in I&I basic science publications for about 15 years. It's got actually a beautiful profile, especially the type 2 of the salt-inducible kinases, which is that it's got this broad, when you inhibit, a broad anti-inflammatory profile. For example, a knockdown of both TNF and IL-23 by a single small molecule. But also with that, a pro-resolution phenotype when you inhibit that target. So an increase in IL-10, one can immediately imagine the benefit for something like IBD, downregulating the inflammation, but also bringing up pro-resolution in the gut to restore GI tract integrity. So we're excited about that. That, I think, has turned out to be, even though it's a kinase, kinases outside of oncology are very tough. The challenge is to make sure you get the safety as clean as you can. We're really pleased with the progress there and looking forward to getting a DC. Multiple indications we think about. We start at IBD, Crohn's, and colitis, but certainly are interested in thinking about SYK2 inhibitor as a broad I&I agent. So those are 2 of our programs that I can tell you about.
You've been working in this as a company for quite some time now. And you touched on it a little bit earlier about how things have changed, but how do you refine and evolve your techniques, and what are the benefits, or what are you trying to achieve as you move forward, I guess?
Yeah, I think what's happened at Nimbus over the last 5 or 6 years is a focus. And that focus is an agent that succeeds in a proof of concept trial at phase 2. That's actually where we start when we select targets. And really what fits into refining the tools that get you there is it actually has to make a meaningful difference for that. And so, for example, I mentioned AI. There are AI techniques that can't have any benefit for us now. And I mentioned the ones that really do. And so we start there. I would not refine SBDD anymore if there's no issue on deriving potency. I think we're very good at that. We need that. But I don't know that there's a lot to gain on that. Improving the ability to get in multi-parameter optimization, everything else around drug-like properties, ADMET, we continue to invest in that. I don't know that anybody's made major advances on the side of predicting safety. I know there's a standard set of things people do, but there's an area that's challenging. And then refining our methods to go from a DC to an FIH and run effective trials, running phase 1Bs that are data rich, that inform a phase 2 and whether you should run the phase 2 and what that phase 2 should be, are all areas of focus. Those are some of the areas we continue to refine. One of the things that may not be obvious about Nimbus is our translational medicine group and our clinical development group that focuses exactly on that. How do we get data-rich phase 1Bs to inform our phase 2? So those are some of the areas we focus on to get to successful POCs.
Everybody's talking about AI and machine learning and quantum computing as being something that is really important in instead of something taking 6 years, bringing it down to 2. Do you think that that's realistic? And do you think that there are better ways to do that? Or do you think that some things just should take time anyway?
Everybody who works in drug development would like to take the industry from 6 years to clinical trials to 2. I don't see it. I sit on the side of skepticism. It is worth saying that Nimbus is not a platform company. I think that with the platform companies comes the need to at least make certain promises. I would say Nimbus sits on the side of being skeptical. We use those products, those platforms, and we assess them as objectively as we can. I would talk less about where things are going to be in 5 years, about what we have today, and I don't see it. Let me say this about it. 6 years is hard. I don't know if that's really the problem. I think the problem is the challenge we should see in front of us is tackling the biggest problems. And I can give you an example of what I mean. I'll talk about 2 big successes to give you an example. I look at Amgen and Mirati and their success on drugging KRAS. Fantastic work. And KRAS had not been drugged for over 30 years, and they thought it never could be. And because it did, the benefits for patients with non-small cell lung cancer is huge. And we'll continue to look at that, and there'll be combinations, but there's a big benefit. They took on one of the biggest problems. As far as I know, AI had no contribution. I look at the drugging of STAT6, same thing. That was Chimera as one of the first companies. It's a real leap forward. I don't know that that is an AI effort. Those are outstanding drug discovery efforts. And I would look to, I think the best place AI can make a difference is to move from working on, if you want to go the fastest, you work on the simplest targets, like kinases. That's not a fair judge. Large pharma and biotech can move very quickly on kinases. They're not necessarily the best targets. Work on the best targets to make the biggest benefit. I think that's where AI should change its focus.
Very good, yeah. What do you see as the future for computational chemistry?
I think it's going to become, and this is another aspect of what Nimbus has done, computational chemistry in large pharma to some extent sat as a separate group from chemistry. We've changed that at Nimbus. They are chemists side by side, and our chemists are computational chemists. That's absolutely essential, I think, to make them fully integrated to make as big a difference. I think the tools will continue to grow in power, and the chemist of the future will use them just very easily. It's just part of the toolbox. If you'd like a parallel, I would bring up where we were with genomics 20 years ago, where people said it was going to revolutionize the business. I was at a company, which was Millennium, that said it was going to improve drug discovery effectiveness tenfold, and it didn't. What it did was provide powerful tools to the biologist. A biologist today would never work without genomics. And that's where I see computational chemistry going, is making the chemist much more effective. But it's the chemist that drives it. And so we're excited about that. And again, side by side with the skepticism of assessing these new tools as objectively as we can.
Flipping that then to your own company, what do you see as the immediate and long-term future there?
I think that we find opportunities across all 3 therapeutic areas where it can make a difference. And I'll mention something as an opportunity for Nimbus, and I see other companies doing the same thing, which is, people will think about first in class. It's an awesome approach, or fast follower, where the goal I think could be add more value beyond first in class. There's a third group, which are targets that have been known for a long time and haven't been solved yet. And people made progress and did not get it into the clinic. We're in an age where the tools in total are more powerful than they were 20 years ago, and people are taking a second look at those same targets. And if it's true that they are as valid or validated as we thought they were, I think you're going to see solutions against those. It's an opportunity for Nimbus, and I think the industry is going in that direction. So that's an exciting place to be. I'll say one last thing about Nimbus that relates to this a little, about the culture. We try to have an environment where everybody is a drug discovery or development scientist across the board. So, for example, we call our new targets process a jamboree. Everybody comes. Everybody proposes targets. Everybody's put up against the same firing line in a collegial way to bring things forward. if you propose a target at Nimbus, you might be directing the future of Nimbus, is the way we think about it. And I think that's exciting. It's really a lot of fun, and is hopefully an attraction to others to want to come join us when we have positions open. At least that's the way we look at it.
So, now you know about computational chemistry, although you might have already. But another really interesting subject and a very enjoyable conversation. It's hard to keep some of these to less than half an hour because there's so much good information. Anyway, that's it for another podcast. I don't have next week's interviews edited, but they are done, so I guess I have to be thankful for that at least. If you have questions or comments or suggestions for guests, please do get in touch, and you can do so at jim@deeptechdigest.com. So, until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast.
The Base to Base Biotech podcast is an Air Coastal Media Limited production.